So this is the kingdom fungi, and we can see here the basal fungi coming down to basidiomycetes, like our core wood rot decay fungi, to the Saccharomyces, so the fungi we like to drink, and then within the remaining ascomycetes we have groups of highly lichenized fungi, but then two big groups of pathogens, the Soderiomycetes and the Dothidiomycetes as well, and nested in between Cochleobolus heterostrophus and Pharenophora Tritici-ripensis here, we've got the Alternaria genus. So this is the Alternaria genus itself, and we've got a lineage of large spored isolates, and then a lineage of small spored isolates. Now, coming down from that very big picture, using these phylogenies have been determined using common phylogenetic loci, but when we get down to the Alternaria alternata species group, which is this group here, we really get down to the limit of resolution that we can use, or that we can determine using one of our standard barcoding loci. And as you can see, there are many morphologically described species, or many characterized species within this group here itself. So we need to try and characterize this a bit better. So biologically, the Alternaria alternata species group are prolific producers of spores, these melanized spores forming characteristic branched chains, and on top of being very common, ubiquitous saprotropes in the environment, we see them as plant pathogens causing leaf spot diseases on a range of hosts. Okay, so traditionally, this wealth of taxa within the species group has been characterized on the basis of spore morphology. And I just show some of the representative diagrams of spore morphologies that I'll be talking about for different taxa. Alternaria alternata is the reference species for the group, and this is typically short spores, very highly branched, whereas we see variants with longer sporophores, such as Alternaria arborescens, which are similarly highly branched, whereas we can have shorter chains without branching, Alternaria geisen, and Alternaria tenuissima typically forming these very long chains without branching. But then this is fine, this captures the diversity of different spore morphologies within the species group. But when you've got over 100 different described species, you're trying to tell the difference between this characterized morphology and this morphology, and some may raise questions as to the validity of these as different morphological species. So of particular interest, I mean, it's all well and good saying this is a species group that needs further characterization, but this group has economic importance too. As plant pathogens, particular diseases, apple leaf spot and pear leaf spot, cause significant economic damage on apple and pear production in their native ranges in East Asia and in North America as well. And with increased movement of plant material, we're seeing risk of establishment in Europe and the UK as well. So it comes down to local screening bodies, such as DEFRA in the UK, to perform screening, and they rely upon fast and accurate diagnostics on which to make decisions on whether to accept or reject shipments which may be potentially contaminated with Alternaria. And essentially what they're trying to identify is what is the difference between an isolate which may be a host-specific plant pathogen, such as the Alternaria meli or Alternaria geisen, and what may be otherwise a ubiquitous Alternaria saprophyte, which is causing an opportunistic rot infection or opportunistic infection of plant material as it enters. So trying to determine what are these host-specific pathotypes from Alternaria which may already be present in the UK. So So to address and further characterise the Alternaria species group, we asked 4 main questions. So using phylogenetics to try and characterise different lineages within the species group, to look at spore morphology and identify whether our phylogenetic clades are supported by spore morphology, to look at the basis of pathogenicity, and also to ask some broader questions as to whether we can observe potential sexuality within the species group, as this may alter potentially how we manage and control this species. Okay, so I can talk you through it. We started off by taking our culture collection of 116 isolates, and through sequencing across 6 loci, we managed to construct a concatenated phylogeny, Bayesian phylogeny, for 5 of these loci. So where I said before that the phylogenies which I showed were really at the limit of resolution using commonly used barcoding loci, these are 2 highly variable loci that have already been characterised in the literature, plus 3 highly variable loci which we developed in-house as well. And I show L152 as an additional locus which we sequenced, which isn't shown here for these isolates. So across this phylogeny, we were able to identify 3 major lineages within the species group, and through placement of reference isolates within our culture collection, we were able to associate these with these different lineages. So Alternaria arborescens we observed within lineage 1. Within lineage 2 contained Alternaria tenuissima, Alternaria mali, and Alternaria alternata. And lineage 3 just contained 3 isolates, including the reference isolate for Alternaria geisen, the pair pathogen as well. So of significance already, we're seeing 3 major lineages falling out, and a clustering of multiple morphologically described taxa within a single lineage as well. So possibly raising questions over their validity as distinct taxa. So having identified these lineages, we wanted to further characterise these to see if they're supported by spore morphology as well. So to do this, we took our isolates and we grew them under standard culturing conditions, using 3D sporulation patterns, characters on growth of the colonies on the plates, and also looking at septation within spores, the actual size dimensions of length and width of spores, as well as additional characters such as presence of branching at different points within the spores, and chain length and branching within chains as well. We were able to put this together and construct a morphological dendrogram from which we could look at associations and clustering of different spore morphologies with each other. So with our constructed dendrogram, we were able to identify major clades within the dendrogram, set at an arbitrary similarity length of 95%. So the spore characters were 95% similar to one another, summarising 95% of the variation within the data set. And within these different clades that we identified, we were able to relate these to the phylogenetic clades that we'd identified in the previous slide. So we can already see that our morphological clades are associated with generally a single phylogenetic clade as well. So these are largely from clade 2, these are largely from clade 1, and you see the blocking down here as well. So this shows that generally our spore morphologies are clustering in a way that's associated with our phylogenetic data as well. But then further to this, we didn't just want to identify whether our isolates were grouping on spore morphology. We wanted to see what's the basis of separation within between these groups. So what are the morphological characters which are making this group B separate from A? So to do this, we use principal components analysis on the dataset. And using principal components analysis, we could pull apart different associations of spore morphology, or different patterns of spore morphologies, which are associated with different clades. And we did this by plotting the residuals of the residuals for each isolate according to a particular principal component. So this is separating out our isolates on the basis of principal component 2 on this axis and principal component 3 on this axis. And just taking principal component 2 as an example at the moment, isolates which score positively for principal component 2, as we can see by the weightings that we have attributed to particular components here, typically have certain morphologies. So we can say that an isolate that scores positively for principal component 2 is typically apically branched, has large colonies, has smaller and shorter spores, and has more spores in total than an isolate that scores negatively for principal component 2. And as we can see, this seems to be separating out different morphological clades within our morphological dendrogram. So doing this, we can say that a clade that scores up here for principal component 2 may be associated with these morphological characters, and these morphological characters we can associate with a described morphology, species morphology, which we determined in this case would be Alternaria arborescens, and in this case would be Alternaria tenuissima. So through generating these associations of spore morphologies with particular morphological clades, or with particular morphological species, we're then able to plot this back on our molecular phylogeny using our 5 barcoding loci that I showed you earlier. So within clade 1, we see almost all of our isolates score positively for an Alternaria arborescens morphology, with very few having Alternaria alternata or Alternaria tenuissima-like morphologies. And the reverse is true for the tenuissima lineage as well, with most isolates here having Alternaria alternata or Alternaria tenuissima-like morphologies. Now the significance of all of this is that all of these morphological characters were taken blind as well. So where in previous studies people have tried to associate phylogenetic lineages within the Alternaria alternata with a particular spore morphology, they've just taken an isolate and said this isolate looks like Alternaria arborescens, this isolate looks like Alternaria tenuissima. And where you have this huge overlap in spore morphology, it's very hard to generate robust data that's associated with that. So by taking multivariate data blind, this has actually turned out to be a much more powerful way of assessing association of morphology with different lineages. So having characterized the Alternaria alternata species group, we looked forward to look at the basis of pathogenicity. Now, I'm going to have to give you a little bit more background on this. An isolate of Alternaria alternata may not or may carry an additional chromosome associated with the production of a host-specific toxin. So in the case of apple, it may carry an additional conditionally dispensable chromosome which allows production of an apple toxin. Whereas the pear toxin, or where the pear pathotype can produce a pear toxin, which is encoded by a distinct conditionally dispensable chromosome, which may or may not be present within its genome. These conditionally dispensable chromosomes produce these toxins through gene clusters on the chromosome, which encode a series of enzymes which are involved in the secondary metabolism that leads to the production of these toxins. So they don't code directly for a toxin. It's a suite of genes which allow the secondary metabolism of either the apple, the pear, or a number of different toxin genes. And thankfully, these have been relatively well understood. This is an example of a region of the apple toxin chromosome. So to date, 17 genes have been identified through disruption studies as being involved in toxin synthesis. Due to primers being available for AMT1 and AMT2, we were able to look for the presence of these genes within our isolates. So where we see presence, we can relate this to our phylogeny. And we observed that within phylogenetic lineage 1, no isolates show the presence of both toxin genes. Within lineage 3, all isolates show the presence of both pair toxin genes. And within lineage 2, isolates are present that show the presence of both apple toxin genes. Now, I know that this looks a bit of a mess. The toxin primers that we got from the previous study, we found typically produce a lot of secondary banding. So this gave us a good indication of isolates that carry toxin genes, but we still see a lot of isolates which may show slightly ambiguous results here. So ideally, and what I show you in a minute, is that we need new primers to better characterize these isolates. But of significance is that this group seems to carry pair toxin genes and therefore the pair toxin chromosome. This group carries apple toxin genes and possibly the apple toxin chromosome, whereas this group seems to lack the presence of these toxin genes or the toxin chromosome. So, if this information and presence of conditionally dispensable chromosomes could potentially offer a use for diagnostics for these particular pathotypes, we wanted to confirm that they are indeed associated with pathogenicity. So taking 3 isolates which tested positive for the toxin gene regions and 3 isolates which tested negative, we were able to assess using detached leaf assays their ability to cause disease on susceptible apple varieties. And we found that the ones that test positive do indeed cause disease, and the ones that tested negative were no better than controls. Following this, to further characterize these conditionally dispensable chromosomes within our isolates, we performed whole genome sequencing on 12 different isolates, including isolates from each phylogenetic lineage and isolates from a range of both apple and pear hosts. And this also includes isolates which tested positive and tested negative for the presence of these apple toxin gene regions. Okay, so I showed previously that we had that bat clone which showed the presence of the 17 apple toxin genes within it. So those were all 17 characterized apple toxin genes currently known. And within the pear, strawberry, tangerine, rough lemon, and tomato pathotype, some of the toxin genes also associated with with the specific toxins produced by these pathotypes have also been characterized. So we took the total suite of, I can't say how many, I think there was 30 or so, 40 maybe, genes here, and using blast searches against our assembled genomes, we were able to look for their presence within these genomes. So this is a table of results, and we can see here the toxins that we searched for, or the toxin genes that we searched for, and we can see here the different isolates that they were present in. So these are divided by phylogenetic lineage 1, phylogenetic lineage 2, and phylogenetic lineage 3. And we can see that isolates within phylogenetic lineage 2 tend to test or have isolates that test positive for the apple toxin cluster, and the isolate that we sequenced from lineage 3 tested positive for the pair toxin genes that we searched for, as well as containing homologs to toxin genes from different systems as well within its genome. And then we also see here some possible homology to genes on essential chromosomes within the genome as well. But this was significant as it's allowed us to identify novel regions within the genome from which we could design toxin primers to look for the presence of, to create better primers for the presence of apple and pear toxin diagnostics. So we took AMT3 and AKT3 as novel, as new regions for diagnostics to be performed on, to make use of, as well as taking this interesting gene here, AMT14, which showed presence within our apple toxin-producing isolates, or apple pathotypes, but also showed a homolog within the pear pathotype that we sequenced as well. So we could possibly develop a diagnostic for both, for multiple pathotypes of phytosanitary importance here, which would be very significant for DEFRA. And through looking at the presence within our isolates, we saw a much better, a much clearer picture of presence of these toxin genes within our isolates' genomes. So within lineage 3, we're seeing presence of pair toxin genes as well as the homolog to AMT14. Within phylogenetic lineage 1, we haven't seen the presence of any toxin gene regions. And within phylogenetic lineage 2, we are able to observe presence of the apple pathotype AMT14, as well as its homolog and AMT4. Interestingly as well, we found that 2 isolates from strawberry also tested positive for AKT3 and AMT14. And if we go back to this table here, we can see that AKT3 has a homolog within strawberry as well, which upon sequencing this locus, we were able to identify that match was indeed to the strawberry toxin gene region. So AMT14 could actually be quite a powerful locus that could identify multiple host-specific pathotypes within the Alternaria alternata species group, which would be significant. Okay, so finally, we looked at the possible sexuality within the Alternaria alternata species group. Now, the Alternaria in general, as a genus, has previously been characterized as an asexual genus. It does have some relatives which are sexual. So Luia is the sexual form. However, below a certain stage, the sexual states are unknown within the species group. However, interestingly, mating type 1 and mating type 2 loci have been observed within a number of taxa within the genus, including Alternaria tenuissima. Now, the significance of the presence of these loci is that fungi typically have 2 mating types. You can be mating type 1 or mating type 2, and you need an interaction between isolates that are mating type 1 and mating type 2 before a sexual interaction can occur. Now, isolates may either have one of these loci or the other, or they may have both, and therefore be able to self. However, it's simple in Alternaria, similar to this system here, where an isolate just contains is heterophallic, meaning it contains either mating type 1 or mating type 2, and it's determined by a single gene within the genome. So, using primers for the presence of mating type 1 and mating type 2, we're able to characterize our isolates. And for through associating this with our phylogeny, we're able to see that within lineage 1, we see, well, within both lineage 1 and lineage 2, we see a relatively even distribution of mating type 1 and mating type 2 loci within these lineages for what is generally considered as an asexual species, and having been asexual from a very basal state. Now what this shows at a minimum is that we must have had recombination, or at least genetic exchange occurring at the formation of these lineages for them to contain multiple mating types rather than a single mating type, as would be expected from an asexual clonal species. So we can say that the Alternaria alternatus species group, if it isn't currently sexual, must have at least had a recent sexual ancestor. Now this is significant potentially in how you could go about managing the Alternaria species group as well. So to further investigate this, we took our genome sequence data and we predicted genes within the genome using evidence from RNA-seq to look for evidence for particular gene models which are being expressed in culture. So from generating RNA-seq data, we're able to align this data to the genome, look for the presence of where we see alignments to the genome. This is evidence for exons. Where we see a read that's broken over a region, this allows us to predict introns over that region. And using this gives us hints from which we can predict genes. And using our predicted gene models for our isolates, we're able to look for the presence of genes associated with meiotic recombination within the genome to look whether they're conserved. Now, meiotic recombination is a well-studied, well-characterized series of events, and is required for a cell to undergo meiosis, as having undergone a recombination event, the chromatids or chromosomes are aligned in a way in which allow them to be pulled apart by the spindle. So recombination is required for meiosis to occur. And the genes which are associated with the process have been relatively well characterized, and total to approximately 80 genes which are involved in this event. So through looking for their presence within our genomes, we're able to characterize or able to identify their presence in our Alternaria isolates that we sequenced. A nearby related Alternaria, Alternaria brassicicola, which causes disease on brassica, as well as looking in related sexual species, Dictydiomycetes, such as Pyronophora triticirripensis and Cochleobus heterostrophus. And we find that the core meiotic genes, so the core genes involved in meiotic recombination, and meiosis-specific genes, which are genes which are only expressed during recombination, are conserved within our genomes, as well as a large number of other genes which have been identified as being involved in meiotic recombination. And we don't see any real difference between the sexual species and our asexual Alternaria here. So this again isn't evidence that Alternaria are undergoing sexuality, but it shows that Alternaria have a potential for sexuality. So, in summary of the work that I presented, I can produce this cartoon. So within the Alternaria alternatus species group, all of our isolates show a single haplotype for the fungal barcoding locus, ITS. So they're indistinguishable in this respect. So the characterization of this species group requires more highly variable loci. So to do this, we performed phylogenetics using 2 highly variable loci which had previously been identified in the literature, as well as 3 loci that we developed in-house as well. Using our phylogeny, we were able to identify 3 major lineages within the species group, and through association of sporulation characters which we collected under blind conditions, we were able to associate these lineages with particular spore morphologies characterized as Alternaria arborescens, Alternaria tenuissima-like, and Alternaria geissem-like. Through looking for the presence of toxin gene regions which are involved in the production of toxin genes within our genome, we were able to identify that isolates within this lineage showed presence of both apple and strawberry conditionally dispensable chromosomes, and isolates within lineage 3 showed presence of pair conditionally dispensable chromosomes. Of significance, though, is that isolates, not all of our isolates within lineage 2 carried these conditionally dispensable chromosomes. Many of our isolates tested negative. So that means that you can't use essential genes on essential chromosomes or loci on essential chromosomes as a basis for developing diagnostics. So if we want to develop diagnostics for these particular pathotypes, we'll have to rely upon regions within these conditionally dispensable chromosomes. Also of note, it's been shown in the literature that an isolate within which I can identify as being present within lineage 1 has been reported as carrying a tomato toxin chromosome. So it means that, well, this gives evidence that potentially all of these lineages can carry the conditionally dispensable chromosomes, allowing an isolate to be a pathotype. However, the different pathotypes may be different between the different chromosomes or the different lineages that they're present in. And finally, we've identified potential for sexuality within the Alternaria alternata species group through looking for presence of mating type 1 and mating type 2 loci, and identifying that they must have at least a recent a recent sexual ancestor or undergoing recent genetic exchange. And through looking for the presence of meiosis-specific genes within our genomes, we're able to identify that Alternaria appear to have the molecular machinery associated with meiotic recombination, and this has been conserved in what's typically been considered an asexual species. Okay. So I'd just like to thank the supervisors that I had while I was doing the project. It started off being a project set up by Srinivas Prasad, who was working at the University of Warwick, however is now at the University of Bedfordshire, and Charles Lane at York. So they originally got the funding through FERA and from a grant from Europe. Upon Prasad leaving, Des Barbara took over the project as principal supervisor. Unfortunately, he passed away, and Charles Lane was promoted. So my supervisors, John Clarkson and James Woodall, had significant shaping of the project at the end to what it was. And during this period, I had a large amount of advice, guidance, and cultures from not only in the UK, but from Russia and the US as well. Yeah, thank you for listening.